Bom dia, pessoal. Então, um prazer estar aqui hoje para apresentar, né, a professora doutora Katarina Emelie, vou tentar falar Svahn Leão. A professora Katarina, ela é professora adjunta no Instituto do Cérebro, na Universidade Federal do Rio Grande do Norte, em Natal. É coordenadora do Laboratório de Audição e Atividade Neuronal, e ela fez o doutorado em neurociência na Austrália e pós-doutorado na Suécia. E hoje, então, a gente vai conhecer um pouquinho, né, mais do trabalho da professora Katarina. Estou bem entusiasmada, fui dar uma olhadinha, né, no que ela trabalha, então acho que a gente vai poder escutar algo sobre audição hoje que eu tenho certeza que vamos todos aprender bastante. Muito obrigada, professora Katarina, pelo aceite. A palavra é toda sua.
Muito obrigada, Patrícia. Então, vou compartilhar aqui. E você fala para mim se tem algum problema, se não...
Tudo certo.
Perfeito. Então, muito obrigada pelo convite de participar nesse simpósio. Estou bem feliz e espero que eu possa visitar no futuro e realmente conhecer lá. Hoje eu vou falar sobre a fisiologia do tinnitus, ou zumbido, e alterações eletrofisiológicas de neurônios corticais. E eu vou começar só apresentar meu grupo. No nosso laboratório de adição e atividade neuronal, eu tenho 2 alunos de mestrado, a Elton e Rodolfo, e 2 alunas de doutorado, a Bárbara e a Ingrid, e um pós-doutorado, Tawan, que também fez doutorado comigo. Então, no laboratório de audição, é bom ter pessoas de fonoaudiologia e também de biomedicina. E o Rodolfo, ele não programa, ele é a pessoa que faz comportamento, então não é tudo como você acha que é, e ele é ótimo também. Mas nessa palestra eu vou principalmente falar sobre o trabalho da Ingrid, e aqui você dá para ver que ela sempre fica no laboratório, não? Não, só que ela faltou na outra foto. E eu vou fazer minha palestra em inglês, mas vocês podem fazer perguntas em português. Só que eu estou na Suécia agora, e meu português está meio travando quando eu falo ciências, então vou mudar para inglês agora. So, tinnitus. I presume that some of you might even have tinnitus, maybe not chronic, but sometimes tinnitus is this beep or some kind of sound that you hear inside your head, basically, and it has no external source. So this can be very annoying, that your brain is making some kind of activity that you perceive as it would be a sound in your surrounding, but there's no way that you can turn this off. It's constantly there if you have chronic tinnitus, for example. And many cases of tinnitus are associated with actually hearing loss. That was the main reason why people believed that there was tinnitus before, that there was always some kind of an auditory trauma, that you had listened to something very loud and that had destroyed part of your the frequencies that you could hear. So some kind of partial hearing loss would then cause this over activity that creates tinnitus. But now we know that it's enough to have noise exposure, but you don't really have to have something causing a partial deafness. It's enough to have a noise exposure very loud for a brief moment. And just to highlight here, this was very recently published, that there's a lot of tinnitus with normal audiograms, that you don't necessarily need to have any hearing deficit. So this is changing the field of tinnitus, because people are realizing that we cannot do animal models where we really cause a loud auditory trauma. We might have to have research models where we just have an increase in sound or noise for a while, and that's enough to create tinnitus. And that's actually what my research group is dealing with. We are not dealing with research models of hearing loss, because we don't want to mix these two conditions. We just want to have increased activity for a moment that will then cause this persistent change somewhere in the auditory system, leading to tinnitus perception. So as I pointed out, that young individuals are starting to say that they have tinnitus, and this was not that much shown in the literature before either. But here are two examples. This is a very large study from the United States, more than 3,000 participants between 12 to 17 years old. And here they reported that almost 5% of them already have chronic tinnitus. That was shocking, because usually in the general population, we have maybe 10 to 15% of people presenting some form of tinnitus, but it's more common in the older population. But here they're showing that this almost teenagers are having chronic tinnitus already. And then in the same year, a group from Belgium actually went ahead and did a similar study. So they sent out these questionnaires, and they found that among these people, there were also 3,000 to 4,000, mean age 16, that 18% had chronic tinnitus. And that's very high. And here, as they put in the title, beliefs towards noise and hearing protection in adolescence, they were actually criticizing, saying that these students that they questioned, they were not that concerned about protecting their ears, about being careful when they go to a concert, for example, to use earplugs, or to not have too loud when you have these plug-in earphones, for example. So this is clearly something that's increasing in the world, unfortunately. So the scary thing with tinnitus, the zumbido pathophysiology, is that there is no cure. There's no way to if you have a chronic tinnitus, it's very rare that it will disappear. Of course, there are some examples, and there are many forms of tinnitus. They can be caused by other diseases, and it can be jaw misalignment or local tumors. There's many forms of tinnitus, but these major ones of hearing loss or noise exposure, or as I call it, noise-induced tinnitus, for these ones, there is not really any cure yet. And I think that's what scares people when they start to perceive tinnitus and they go to the clinician a few days or a week later, and then there's some person there saying like, yeah, sorry, you're going to have to live with this. But of course, there's a way to treat tinnitus. But this is mainly focusing on alleviating symptoms. So what are symptoms? For example, it can be there is a difficulty to hear or to focus. If you're standing far away from someone and you have this beep in your head, it might be hard to actually hear what someone a bit further away is like trying to tell you, because you have this internal loud noise in your head that just makes it difficult to focus and hear. And you can imagine that this can cause bad mood. It's irritating to have this loud noise that you cannot do anything about. And that can be very stressful. And when you're stressed, and also when you're stressed about, will I be able to perform this particular task? Can I go to a job interview? Will I hear what they're asking me? That leads to, obviously, to forms of anxiety. And when you're starting to feel anxiety about being able to live your normal life, being able to study or being able to perform your work, then depression is often linked to it. So some studies say that about 30% of people with tinnitus have depression, and some even say that up to 50% have some form of depressive disorder or symptoms. So all of this together obviously can decrease quality of life. So tinnitus will not make you die earlier or like be life-threatening in that sense, but decreasing the quality of life and depression, we all know, is very deliberating in our culture. It's really one of the most rising health problems that we have. So in that sense, it is very important to understand this condition much better. But then looking at some treatments, one of the most successful treatments is cognitive behavioral therapy, for you to accept and understand that you have this phantom sound perception, that you can live with it and you can still do everything you want to do in life. And mindfulness comes very close to this as well. You can meditate, you can turn the sound you're hearing into something meaningful, or some kind of a maybe it will help you to realize, oh, I'm very stressed because my tinnitus is louder right now, so I have to actually use this as my body is telling me that I need to slow down and relax, for example. And this is all very beautiful, but you can imagine if you are in a bad mood, stressed, and feel anxiety, it might be hard to practice cognitive behavioral therapy or mindfulness. Sound masking is a different approach, where you can actually have a little device, plug it in your ear, and it will play some kind of sound, and it's meant to decrease the perception of your tinnitus pitch. So for example, you can have a hearing aid that will play waves or rain or this kind of general just of a different frequency than what your tinnitus is perceived in. So it can kind of block it, and when you have a sound that's blocking the other sound, then you know that this one you can turn off, this is like under your control, then you relax a little bit more, you know. So that's effective, but not to all patients, but to some. Then there are indications of way to actually treat the neurons, the actual problem. For that, we have transcranial magnetic stimulation or transcranial direct current stimulation. So this is just two titles of two large reviews, but it's saying, for example, that repetitive transcranial magnetic stimulation has a possible efficacy if you stimulate in a certain way, in a certain place, to treat tinnitus and also auditory hallucinations. So that's interesting. Possible efficacy actually points to the fact that maybe in a study with 20 patients, 5 of them had some kind of relief, or they heard it less, and so on. So it seems that some patients are helped by this, but not all, unfortunately. So there's no standardized way of treating patients, but there's hope that there's some kind of approach that you could actually try to treat it. And if you do this repetitive transcranial magnetic stimulation, you can do it for several weeks in a row, and that could potentially decrease your tinnitus perception. More recently, there was another large review from the same consortium looking at this direct current stimulation. This is when you actually put electrodes onto your head, create some kind of dipole, and you have current spreading across your scalp. And this could, in theory, affect neurons on the very surface of the cortex. But here they show probable inefficacy, and there seems to be an absence of clinical effects in tinnitus, unfortunately. So where is tinnitus? So this is just a schematic from a review showing auditory and non-auditor pathways in tinnitus. And just what I want to show you here is that it starts, sound comes into the cochlea, and then it goes to the cochlear nucleus. And this is the region that is most presumed to be the origin of tinnitus, because here we have so much sensory input. So the wind blowing, or you're walking on some ground that's vibrating, all these kind of sensations that couples to your sound. So this is an area that's very plastic, high plasticity. And there's lots of studies showing alterations to neurons in this area. Then the auditory signal goes up into the midbrain or inferior colliculus. We have some other input from the cerebellar pathway going to the thalamus. And here we have input from the hippocampus and amygdala. So here we can have possible memories of sounds, or fear of sound, or aggression, or all this complicated limbic system input. And then we have the primary auditory cortex, and secondary, as well as the associated anterior auditory field. But what I just want to point out here, except for this area being the origin, is this area, which is the one that is close to the surface of the brain, where you could potentially manipulate using transcranial magnetic stimulation. If you notice, this arrow is much thicker, so it's actually representing that we have a lot of feedback from the auditory cortex to the lower structures, and this feedback could potentially reset problems in lower areas. So there's a lot of hope that this kind of strong output into the descending auditory system might be helpful for understanding the mechanisms of tinnitus and maybe even correct it. One should still remember that there are some limitations to transcranial magnetic stimulation. First of all, for a human, you can do these coils, you can create a magnetic field, and you can stimulate. But for a mouse or a rat, these coils are always a little bit too big, because when you make them very small, they might be heating up a lot, and you don't want to burn the animal. So it's a bit problematic to focus it on a small region, for example. And another thing is that when you pulse current to this coil, and it creates a magnetic field. And in your brain, what happens is that this turns into an electrical field. So we have some potential differences. And as you know, the brain is full of neurons with voltage dependent channels. So if you change the electrical field, you might hit the threshold to open certain channels, and we have current flux, we have probably action potentials, and so on. But what happens is that this electric field gets quickly weaker as you enter the brain. So where you will have your strongest effect is always in the very superficial layers of the cortex. And that's important to remember, that we're dealing with the neurons that we reach will be in this particular compartment. So what neurons do we have there, and how are they affected? I mean, this is basically the main research question for this part that my group is working on. It's like, what cortical neurons are altered in tinnitus? So just to finish the rather long introduction here, but this is some previous work where they put an electrode into the brain. They have these animals suffering an auditory trauma on day one, and you can see an increased activity here in the auditory cortex for certain frequencies that you're playing. But what you see is that after 2 days, this increased activity seems to disappear, and it doesn't really stay. So that's a bit of a mystery, because if we have tinnitus that turns chronic, where does it come from? Because here it looks like, all the neurons started firing a lot, but then it decreased rather quickly. So what about the long-term effect? And why can't we really see this in these general electrical recordings? Obviously, we have to go into more detail. So here's another rather recent paper where they looked at this more long-term effect. So they do an auditory trauma here, and they see that the auditory nerve actually decreased its output for the whole week afterwards. But neurons in the cortex, pyramidal cells, And here they're calling it the corticofugal projection. So that means from the cortex downwards to lower structures. Here they could show that these neurons that projects from the auditory cortex to the, in this case, inferior colliculus, they actually had a standard response here. On the day of the trauma, they fired very little, but then afterwards, up to 7 days later, they have an increased output or gain to sound stimulation, meaning that they are probably more excitable. So here we have a certain subtype of pyramidal cells, the ones that project downwards, and they seem to be more excitable, giving a larger output. So there is for sure something happening in the cortex, and many other studies show similar things. But that was just an example of the pyramidal cells. And here's another example where we kind of get an idea that the interneurons may also be part in tinnitus. So this was a study from last year, and they looked at this classic thing that the cortical maps, the frequency maps are rearranged after when you have tinnitus due to the hearing loss. And here they showed in transgenic mice and with some specific cool techniques that it's not. It's actually the maps remain, but what changes is this enzyme that we need to make GABA. That decreased 10 days after an auditory trauma. So the interneuron seems to be less active here in general. So this is something we also have to keep in mind, that we also might have less interneurons active, or do we have more pyramidal cells active? These are things we don't really know. So now getting to the study here of my student, Ingrid Nogueira. She actually did this during her master's, and she's now starting her PhD. And we pre-printed this in bioarchives because we needed to show this in for a grant application, but it's already now in submission, and I think we're going to get it out just now before the end of or hopefully get it accepted soon, now in the beginning of the next year. So Ingrid, she studied loud noise exposure differentially affects subpopulations of auditory cortex pyramidal cells. So what Ingrid did was that she submitted mice to noise exposure, as illustrated here by the speaker. And 7 days later, these animals were sacrificed, and she made slices, sections of the brain to target the primary auditory cortex, and recorded electrophysiological properties of these layer 5 pyramidal cells. And from the literature, we know that you can divide them into subtypes, and 2 main subtypes, A and B, have been shown in other papers to correlate with the type A to be these feedback neurons that project to lower structures, while these type B cells are going to the contralateral auditory cortex. So they are more intra or corticocallosal. And the way that she separated these cells is that she made sure to only record from adult animals, and she had to make some special solutions to keep the slices healthy. And then you can look at simple action potential firing. And in the end of a voltage or a current clamp step, you actually see this after hyperpolarization. And if you instead give a negative current injection, you will see this hyperpolarization in response. And when you stop it, it usually gives this after depolarization. And this is dependent on voltage-gated ion channels that type B cells don't really seem to have, because you don't get this overshoot or undershoot. So it's a simple way you record, you make your standard recordings, and then after you can separate them into 2 classes. And that's what Ingrid did. And she recorded a bunch of electrophysiological properties, analyzing them in control animals and noise-exposed animals. And she divided them into type A or type B cells. And then she started comparing them. And we didn't really see many major differences. There were obvious differences between type A and type B cells, but between control and noise-exposed type A cells, they were very similar, but there were slight little differences. Except for one value that I'm going to show you in the next slide. But what we did, just to make sure that we're seeing the real picture here, we took the help from our statistician, Tiago, and he did principal component analysis. That means that you use all these 16, well, in total it was 16 values or electrophysiological parameters. And then you do these calculations to see how much the variability of a certain group of parameters is, or how much certain parameters contribute to the variability of the data set. And what we saw here, not surprising, was that type A and type B cells, they were different if you plot in a 3D plot principal component 1, 2, and 3. That stands for 60% of the variability of the cells, meaning how they are electrophysiologically different. So we could see that type B cells clustered here, type A cells clustered here in light blue. And then in light pink and red, these are after noise exposure for the two conditions. And then we could see that they kind of get further away from each other. They get more different. So that was interesting. We see some kind of trend that these cells will be electrophysiologically more different from each other. So what does that tell us? It tells us that after noise exposure, the neurons of the cortex here in layer 5, they are not changing in the same way. They're actually changing in different ways, depending on their inherent properties. So a noise trauma does not affect all cells equally. And that's important to understand. So here, just focusing on firing frequency, a very basic measurement for electrophysiology. And here it was interesting to see that type A cells in controlled conditions and type B cells in controlled conditions, they are quite similar usually. That's why that's not really used to separate them in different classes, because they're very similar. But after noise exposure, for both of these two types, we could see that there was some kind of change here in the initial frequency, the first two spikes, and in the what we call steady state frequency, in the end of a 1 second train of firing. So just picking out the data here from 200 picoamps from these two plots, and we're showing them here in the bar graphs instead. So what you can see here is that the initial frequency of type A pyramidal cells decrease after noise trauma. And here's a typical example of that. For type B cells, there was no difference. But then instead, if you look at the steady state frequency, we saw that it was again decreased for type A cells following the noise trauma, but increased for type B cells following the noise trauma. So that really surprised us, that we had like different or opposite directions of how a noise trauma would change a basic parameter as firing frequency. So we went on to see if there was a difference in gain, similar to this other study in vivo that I showed you in the introduction, where they saw that the cells managed to respond stronger after noise trauma. So here we just simply give them different current steps and see how much they increase the firing frequency in response to 150, 200, or 200, 250 picoamps. And then we just fit a line to that, and that becomes the slope of this line gives us the gain, so hertz per picoamps. And we did not see any change in the initial firing frequency. And for the steady state, we didn't find any significant decrease or increase, but we could see that after the noise trauma, these two cells that usually were similar in how they would increase their firing in response to input, now became significantly different. That one is less likely to increase their firing and the other one more likely. So there is something there showing us that these two cell types are becoming different. So now what we're currently doing and is actually increasing this study to study also in vivo. So for that, we are teaming up with our the neighbor lab, Martinoti Leão and Jessica Winnie, who is an extremely talented PhD student. So she's doing implantations of these miniscopes, miniature microscopes. So what she does is that she actually implants a small lens into the brain, a very, very small lens. So she makes like a little window, and then she places this lens here, and then she locks it in place with dental cement, this acrylic, and then she has a little head plate there. And onto this head plate, you can then, when you do experiments, connect this mini microscope. And these miniscopes are extremely lightweight. They are basically the thinnest plastic you can imagine, and then there's a little chip on it, and you have a little optic fiber to pick up fluorescence. So using genetically encoded calcium indicators, we previously obviously injected that into layer 5 of the cortex, and then on the day of the experiment you can actually image cells live in the awake animal while it's listening to tones. So these gray bars here are tones. And then the absolutely fantastic thing with this technique is that you can image cells and then you can see how certain cells, like you pick up cell 1 and 2 in this example, and see how they respond to sound with increase in calcium. And then 1 week later, you can image the same cell, because based on how they are, the anatomy of these cells, you can, using programs to statistically recognize, was this the cell that we recorded from before? Yeah, because it's close to this one and close to that one and close to this one and so on. So you can find the same cell 1 week later and see how it's firing then to the same kind of stimuli. So this was obviously after the same kind of sound trauma that Ingrid did to her animals, which is not that loud. It's 90 decibels for 1.5 hours of broadband noise. That's what we're doing to them. So after 1 week of this loud noise, we could see that the black cell here actually fires much more afterwards. So that could possibly be the type B cell that we saw in the slice recordings. While here in red, we see a cell that fires less now in response to the same sound. So that could be a possible type A cell. So this is very interesting, and that's something we're now increasing our focus into identifying and seeing how this is, if this is truly happening in vivo as well. So now, just to come towards the end, I also said that there might be interneurons that are affected by tinnitus. So here is actually a former PhD student of mine, Marcus Hilscher, my first student. And he did a study, but not after doing this noise exposure. These were just control or normal mice. But he also looked in slices, and he was focusing on an interneuron that's called a Martinoti cell. This Martinoti cell has the cell body in layer 5 of the auditory cortex, or all throughout the cortex. And then its axons goes all the way to layer 1, where it bifurcates a lot. And here it can actually inhibit the distal dendrites of large pyramidal cells. And what Marcus found was that it preferably connects to type A pyramidal cells. And it seems to bypass the type B cells that are close by. It focuses on the type A cells, he found. And as they have this possibility to inhibit the distal dendrite, they kind of regulate how these neurons are firing. So after testing a lot of different frequency, Marcus found that if he optogenetically stimulated Martinoti cells, so the whole population of Martinoti cells in this area, using a genetic marker and injecting channel rhodopsin into them, then he could flash blue light at a certain frequencies, and we found that little bursts of activity was the best frequency. So 15 hertz in 2 hertz bursts. If he did that after he had patched 2 pyramidal cells, and they were firing randomly, so we have a gray trace and a black trace, just showing that they're firing here and there like pyramidal cells do. But then when he turned on the light, he could see that after a relatively short time, these 2 neurons started to synchronize, and synchronize extremely nicely. So that shows that the Martinoti cells have a capability of synchronizing the output of the type A cells. So we know that, and now we wanted to understand how the type A cells are. But it was surprising to us that the type A cells were firing less, and we wanted to know, but is that because of maybe the Martinoti cells? So Ingrid has started some preliminary data, and now she's patching Martinoti cells in noise-exposed animals. And now we have expanded because there seem to be different subtypes of Martinoti cells. But basically in this most prominent subtype of Martinoti cells that Mark studied, preliminary data shows that they are, after the noise, they are harder to excite. They need more current to generate an action potential, which is classically an old-fashioned named rheobase. And she also showed that after noise exposure, they had a lower threshold for generating an action potential, meaning that if this threshold for generating an action potential is closer to the resting membrane potential, then they're easier to excite. So very briefly, the preliminary data shows that Martinoti cells seems to be more excitable. So maybe that's what they're doing to the type A cells. So finally, just trying to put this together in some kind of model, what we have seen is that we have this downwards projecting type A cells. They seem to fire less. And the type B cells that project contralaterally, they seem to fire more. And the Martinoti cells that are specifically inhibiting type A cells, they seem to be more excitable. So then that all the time fitted with this idea that, okay, we have decreased feedback out through the auditory, to the descending or to the lower auditory pathway. But then this other paper that I showed you in the introduction, they saw that there were more output. So now we have added this idea that maybe they fire less, but are they maybe perhaps more synchronous? The few spikes that do come out, are they more synchronized? Because the Martinoti cells are keeping them in order, so to speak. Because then, even if they fire less, if they're synchronized, then the output will be stronger, because several neurons firing together will obviously be more capable of exciting lower structures. So here we have to continue studying this. So we're adding on a question here to whether type B cells, because there are other studies showing this, showing some interesting data. So we are wondering, could type B cells be part of driving the Martinoti cells to inhibit these cells more? Could it be that the type B cells don't locally excite, but could they contralaterally excite Martinoti cells? So that's something that maybe and hopefully Ingrid can study next year, because she's going to do a 1-year PhD sandwich here in Sweden, where I am currently. So then the conclusions of so far of this study is that noise exposure increases type B pyramidal cell firing, but lowers type A pyramidal cell firing. And the questions that I kind of just mentioned to you is that could there be increased interhemispheric connectivity in tinnitus? Do we have decreased feedback, or do we have more synchronous feedback? And are the Martinoti cells truly overactive? And what subtypes in that case of Martinoti cells? So based on these questions, we kind of form new hypotheses then that noise exposure might synchronize type A pyramidal cells to give a more powerful output. So that is something that we could, in theory, study with miniscopes. So continuing that together with Jessica and Hishardson. And that's going to be very interesting. Jessica is showing that she can record from hundreds of cells at the same time using the miniscopes. So it's going to be extremely interesting when we can test this in our mouse model of noise-induced tinnitus. And then, can we increase inhibition to type A pyramidal cells by exciting Martinoti cells? Because, okay, they might be part of synchronizing them, but if we make the Martinoti cells not fire rhythmically, if we make them fire a bit more unorganized or in different frequencies, could we possibly instead just increase inhibition and somehow decrease the activity of type A pyramidal cells? So that's something we want to see. And using transcranial magnetic stimulation, can we test with different frequencies and different intensities of magnetic stimulation? Could we maybe scramble the output of Martinoti cells in some way that they don't make type A cells more rhythmical? So obviously, all of this boils down to this main question: can we decrease tinnitus sensation? But it's really interesting if we can do that using transcranial magnetic stimulation in an animal model. So for that, we have teamed up with George Nascimento from the biochemistry department, and he has actually made us a little coil. He first started testing the coil here and like seeing what size you need to generate a certain magnetic field. So here's this little test coil. And then he made this 8-shaped coil, which is kind of typical. And then he bent it a little bit to increase the focal point. So here's another little test coil just to see if he could focus the magnetic field. And he needs enormous batteries to generate these pulses without generating noise. And right now he's trying to figure out a way to do this in a sense that it doesn't heat up the coil so much. But here is some very preliminary pilot experiment when we try to create this magnetic field over an area close to the auditory cortex of an anesthetized mouse, and without heating up these coils too much. And we were very excited to see that in very, very preliminary data, that just after 20 minutes of rather low frequency transcranial magnetic stimulation, these animals were sacrificed and sliced, and then with the help of Julie Meyer, a very talented student from a neighbor lab, he did CFOS activity, or CFOS immunohistochemistry, and we could see cells here in the probably layer 2 maybe that were lighting up with CFOS. And here is another example. So we were very excited by that short stimulation of very low frequency, but still we could see some cells in the very surface of the cortex lighting up. So of course, we don't know what cells these are and how it's affecting, but it gives us hope that at least transcranial magnetic stimulation can work in mouse. And we are eager to continue this as the pandemic blows over and we can start up experiments again. So just to sum it up for you in an image, this would be to stimulate with TMS to actually generate more liberation of GABA from these inhibitory interneurons, the Martinoti cells, to inhibit the distal dendrites, which usually functions as some kind of regulator of all different types of input to layer 5 pyramidal cells, to inhibit their firing. I'm pointing here to this figure, which is actually just this one traced, but you can imagine the pyramidal cell here next to it. The pyramidal cell would be larger and have a much prettier dendritic tree, but I think it's only me who knows that. So thank you so much for listening. I hope I managed to give this talk in 45 minutes as I was planning. So here, just for you to see our hearing and neuronal activity lab together with our neighbor lab, the neurodynamics unit. And we have lots of incredibly talented students and technicians. And this is Richardson, this is George, always present, helping us with the TMS. And this was Ingrid performing all of this data that you saw. And we are sponsored by, but we have funding from the American Tinnitus Association and Sempic and Capis. And this is our brand new, super fancy building that we are right now moving into. So instead of having these tiny little laboratories, we're going to have better space and better acoustic isolation and so on. And we're very excited to move in there. And I hope that you can maybe come and visit us in the future and maybe be part of our house symposium that we also usually keep towards the end of the year. And also, if you want to come and visit us in Natal, you should go to the beach. We have the beautiful Ponta Negra beach with the Mohoji Careca. Now eu vou voltar para português e falar obrigada por sua atenção. E se você tem perguntas, estou bem feliz de tentar responder essas perguntas. Obrigada.
Muito obrigada, Catarina. Excelente a sua apresentação. Achei assim, eu não entendo nada do tema, mas foi muito, muito didática, né? Fiz várias anotações, tem algumas perguntas, mas eu vou deixar, então, os alunos, né, e quem está nos assistindo, né, fazer as perguntas. Então, se alguém quiser aqui no chat abrir o microfone, ou se quiser, né, escrever, não sei se a gente já tem alguma coisa. O Matheus tem uma pergunta, né, Matheus? Fica à vontade.
Tenho. Não sei se eu pergunto em português ou em inglês, vocês que preferem. First, thank you for your lecture. It was very nice. My question is there any relationship between tinnitus and dementia? Because recently there's a committee that published that 12, yeah, I think so, 12 risk factors in the life course that can be you can change to prevent dementia, and hearing loss is one of these factors. So I would guess that maybe it has some relationship between tinnitus and dementia, but I don't know if it's true.
Yeah, I'm not an expert in dementia, but obviously any kind of sensory deprivation has often been linked to dementia. Even just losing teeth, they say, can increase the risk for Alzheimer's because you don't have any sensation from your mouth and so on. But for tinnitus, as tinnitus is strongly correlated with hearing loss, I presume that people that are old enough to have a age-induced hearing loss, they are also more of a risk factor of having Alzheimer's due to their age. So I do think there's a strong correlation there. But it's not really that form of tinnitus that we are studying. And I can presume that, for example, transcranial magnetic stimulation might be generally good to people with dementia, just to increase activity of the brain or increase metabolism or things like that. But strong correlation with Alzheimer's and tinnitus, I do not know, but age and Alzheimer's and age and tinnitus are correlated.
Thank you very much.
Pessoal, mais alguma pergunta? Se quiserem fazer em português, também pode ficar à vontade, né? Eu até comecei falando em português, então não se intimidem.
Okay, another one. You showed us that in your data that you have some pyramidal cells that can rate in different frequencies. And can I record this pattern in the EEG? I don't know if I can make it in mouse or just in humans.
No, that's a very good point, a very good question. And it's really part of the big problem of when you look at the human brain, is that we cannot pinpoint subtypes of neurons that well in humans. And also an EEG will just give the overall general electricity of the brain. It will not be single neurons firing. So an EEG will just give you the large output. And there's a collaborator with me here in Sweden, actually, why I'm here. He's trying to do these kind of measurements, and it's tricky. So they have actually developed a way to measure magnetic fields. So not stimulating the magnetic fields, but measuring magnetic fields in patients with tinnitus to try to pick out more detailed information. But the machine to do this, I cannot even remember what it's called, but it's I think 2 meters high or something. There's this massive machine that they have on this very fancy institute in Stockholm. So unfortunately, the EEGs that you can make very cheap, you just put this hat with all your electrodes, and they will not give you specific information, but they might still give you indications that you have an increased activity, for example. So that might be interesting to test. But the mouse models are really good because they can pinpoint subtypes, and understanding the electrophysiology of these cells, we can know what kind of stimulation they will respond best to. And then that we can translate back into treatments for patients. It's more about what stimulation frequency, magnitude, polarity, how many repetitions for how many weeks, that kind of information that we aim to bring back to patients and the clinics.
Perfect, thank you.
Katarina, in the same direction of Mateus' questions, I was thinking about the relationship between suicide and tinnitus, because you mentioned that there the anxiety and depression. So I was thinking about if it's something that can make you do something very extremely. And so I don't know if there's a relationship between the two.
Yeah, there is for sure people suiciding due to tinnitus. It's awful. It's really, in that sense, it is a deadly disease. And there is, of course, several pages of that. I think there is some internet forum of people collecting these stories even. It sounds very morbid and I haven't checked it out, but it can be very stressful to certain individuals. It can be extremely stressful, and going to a bad physician, a bad doctor that will just say, learn to live with it, that can be really horrible to a young person specifically, to hear that there's nothing you can do about it. And I have, unfortunately, personal experience in my family. My sister has tinnitus, and she has always been deaf on one ear, but now she has, since 2 years back, she got tinnitus, which is terrible since I studied it. And I was like, no. And she went to a doctor like that and came home crying, and hers is stress-induced and has to do with a very stressful job, and it comes and goes. Hers is different. But I just told her, go to another doctor. It's not true. Go to a tinnitus clinic, as someone who is specific, who understands it. And then she went to another hospital, and they were amazing. They gave her all these different ways of dealing with it and so on. But it's important that we bring that message to people that there is things that you can do about it. And there is a lot of labs in the world like mine trying to improve and to understand the mechanisms better. But I can just say from like having my lab in Natal, and we're just starting to publish on this topic because, you know, it takes time to finish the studies. And I still get emails once or twice a year from people who are desperate, who are begging me to include them in their studies, that cannot deal with it. And they're usually on heavy medication. So I didn't include that in my talk, but people, the simple way is that people get anxiolytics medications and antidepressant medications. And they will not treat your tinnitus. They will just give you side effects that might be even worse. So if you take a lot of anxiolytic medication, you will be like low and you cannot perform in the way you want to. So it's actually recommended against by proper tinnitus clinicians to just give antidepressants or anxiolytics. That's not how you should treat it. But of course, it's the easy way. But people are very stressed about it, and I hear them. And there's an excellent clinic in São Paulo where they have a team of otorhinos, how do you call them? Yeah, and people doing acupuncture, checking they're doing everything, and all the auditory tests, all these things, you know, and try to make individual treatment plans for each patient that they get. And I know there's in another part of Sao Paulo, they also have a similar approach. They have this massive team, and they all go in and assess and try to figure out what type of tinnitus a certain patient have and what would be the best treatment for that one. So it's starting, but it's taking time. And when people are not even knowing about it, you know, they can be stressed. So if anyone in the audience have tinnitus and feel very stressed or insecure about that, they can send me an email, and I can indicate you to these web pages and labs and give you a little bit of hope, because it's awful that suicide actually exists for something that
I could
should
imagine.
not.
Especially when you mentioned that that is increasing between adolescents, and you know, so of course I think about probably is related to the lifestyle. I imagine it because of the loud sounds, and it is why the other question that I have is about the noise exposure that you submit your animals. So how you do that, how you choose the frequency, and what's the kind of the noise, and also I was thinking about that if you can translate this to humans, what kind of sound, or I don't know how you measure that in the lab, if you can translate this to humans or not.
Right. So first, just a quick comment. We are actually doing a study on 240 high school students from Natal to see if they have tinnitus. So we're starting this more reaching out to the general public in Natal. And just randomly there, we found it was not that many. It was maybe 4 out of 240 that had chronic tinnitus, but still. So we want to, in the future, also educate about this, you know. Obviously, modern day earphones are these ones that you plug into your ear, so you block all the other noises, and then you really might listen very loudly to sound without noticing it. And that's a problem, and we see that in the young population. So during my PhD, I studied both deaf mice and normal mice. And during my PhD, I submitted mice to 75 decibels, which is loud, but not very loud. And I saw there were changes on ion channels. So I know that the auditory system is it regulates quickly, just within the hour. So then now when looking at tinnitus, we are using 90 decibels, which would be similar to very loud traffic. So noise in your surrounding, like cars driving by and whatever, lawnmowers or these kind of things. So 90 is loud, but it's not that it destroys your ears. It's just like it's annoying, you know? So we do this for 1 hour or 1.5 hours to these mice, and we have models when the mice are awake or the mice are anesthetized. And then the important thing we do is that after, we leave them for 1.5 hours in silence. And complete silence is also unusual for the auditory system because it's hardly never silent. So other papers have shown that if you do that, it apparently worsens the tinnitus. You have a higher risk of developing tinnitus. So that's something I find interesting, and maybe something one can think about for humans also, that usually what a young person might do is they go to a street festival or a concert, and they're very bad speakers. They're usually terrible quality, and that sound is very, very loud because the people that runs these shows they usually have hearing loss themselves, so they don't hear how loud it is, and this is a reality. So they go to these shows and they don't want to be silly in front of their friends, so they're like, yeah, no, I'll go there, you know. And then they go home and they have this like in their head, probably this temporary tinnitus. And the natural reaction to most people is that, okay, now I have to make everything quiet so I can relax my ears. That might not be right, because maybe you need a little bit of sound just to keep the metabolism going, so you can clean out things that were broken or excitotoxicity. So maybe have like the radio on, but on a low level during the night, just to keep the mechanisms of the neurons, the cleaning of the ears, you know, like cleaning not of the ears, but of the neurons. So there's a lot of misconceptions. So a lot of people with tinnitus, they strive to like make it as silent as possible, and that's obviously impossible if you have tinnitus. So I'm trying to tell people, no, it doesn't have to be silent. You're not helping the brain by keeping it absolutely silent, because that's unusual too. But low music or this kind of, you can play it on YouTube, nature sounds or waves or something like that. That's actually good for you, and it decreases your stress. So yeah, I hope to write a popular article about this soon, where I can give some scientific.
Yeah, it would be very helpful. Yeah, for sure, will help a lot of people. That, you know, for me is a novelty, and I learn a lot with you. And I just think about it. You mentioned at the beginning the wave sounds that can help people, and it makes a lot of sense if you think about it. And I just have another technical question about when you use the mice, you use male and female, and what is the age of the animals?
So we have some studies only with the males, but most studies are both male and female, because obviously the electrophysiological properties are the same for core brain function. We use adult mice, but not that old, because we are currently only using the black 56, you know, the general lab mouse, and that one actually loses hearing with age. So after 3 months, that one will have a already decrease of hearing, so it's not that practical. So we use them we start our experiments at 1 month and we finish at 2 months. So it's a young adult mouse. But we want to now, when we hopefully have a larger animal house, to buy some more specific mouse strains that we can use instead. And it's interesting that all the mice strains that have really good hearing throughout life, they often go blind, while blind mice have instead intact hearing. So it's tricky to use mice. But the black 57 mouse, it's good because it's vulnerable to acoustic trauma. So it's actually good, and it's also the standard mouse for optogenetics that we are using for other studies that I didn't present here. So it's still a good laboratory mouse.
Pessoal, mais alguma pergunta? I have a question, if you can make. I was just curious about the de Nascimento that you mentioned that he was doing the TMS machine. So is he Brazilian?
Yes, George is Brazilian.
A cambiara toda o negócio, eu fiquei imaginando, só podia ser brasileiro, né, para fazer toda aquela coisa de, enfim, eu tinha quase certeza absoluta que era brasileiro, mas só queria confirmar.
But this is truly what I love about Brazilian science, because here in Sweden, people have so much money to buy equipment, and in this corridor, they have everything, but they don't use it because they got it and they didn't find a student who knows how to use the two-focal microscopy or whatever. And in Brazil, we have nothing, and we're like, let's build it. We don't afford to buy it. It's going to take too long to import. Let's make it ourselves. And like especially UFRN or the Brain Institute, we have, I think our group have 5 3D printers. We are printing stuff we need, you know, and we are building stuff we need. And talking to George, he's always like, let me think about it. I can make it, you know. And then he shows up a month later with a prototype, and I'm like, oh, really? So yeah, I love Brazilian science, and I hear this from so many people in the world. They have students from Brazil, and they're just saying that they are amazing students. They're extremely sociable, and Brazil have so poor funding, but they do such good neuroscience. So that's a congratulations to all of you from Europe.
I was just amazed about the miniscopes. Oh, this is really cool, and for sure you can, you know, I'm sure that you're going to find really exciting results if that technique was enabled by that. So nice.
Yeah, and that's really revolutionizing, because before they had to have the head fixed animal with a big microscope, and that it's not very natural to the mouse. And this one, they are so light, you will not believe it. So the mouse is just walking like normal. And I know Jessica, she spent 6 months or maybe a year learning to do the implants. These are tiny little things. You cannot touch them because you blur the lens or you break it. So you have to be extremely detailed with how to implant it. And then you can actually attach a prism to it. So you have your lens, and then you have the prism underneath. You can actually image sideways if you stick it in. So you can stick it in and then image from layer 5 or layer 4. You know, it's amazing. But you have to glue these tiny, tiny, tiny little things together. And then when you implant it, and then later you put the microscope on, of course, and then you can adjust with these tiny little screws the focus. So you focus on the level where you have the most cells expressing these things. And now the miniscope version 4 is out. It's very expensive, but still, you buy it, and that one has a gel, so you can give a current to change the panic while you're pressing on it. So it's booming, this thing, and it's very few people in the world publishing yet, because everyone now is learning it. And I know Jessica, she's in the middle of her PhD. She's already offered postdoc position in Canada. They want her to come. They just heard of this amazing girl that's so talented, and so it's really encouraging. And I'm so happy that I have connections, you know, so I can ride a little bit of the wave. And I hope for my master student to learn this technique in the future.
Yeah, it's really, really nice. So people, more questions? Josiane, queres abrir teu microfone? Tu consegue ler, Catarina?
No, sorry, I don't have my chat open.
Ela está colocando aqui, a Josiane. Não sei, Josi, queres... se forem feitos testes em diferentes tempos de exposição, noite ou dia, se a luz pode influenciar nas ondas e persistência do zumbido.
Excellent question. Excellent. There's this whole group here in Stockholm. They focus on the endocrine, how hormones affect tinnitus, obviously stress hormones, but also the circadian rhythm. And they have very recently, and I think it's just published now, shown that, now I can't remember if it's worse in the night or in the day, but I can't remember because it's very fresh data. But they're showing that it's extremely important that you do your tests at the same time every day, because otherwise you will have variation in your experiments depending on the day. And whether light can affect it, I do not know, but for sure hormones regulated by light can regulate how vulnerable your auditory system is to the trauma. So this is becoming, we're starting to understand this now, so it's extremely interesting. They are using a pharmacological model for tinnitus, this cancer drug, cisplatin, but I would really like to test it. And when I heard about this work, I was so worried, and I was telling my students, oh, you have to make sure you do it at the same time. And then my students are like, I always do it in the morning, one group, and then the other person, like, I always do it 4, and the other person, I always do it at this time. So the students have a natural rhythm for when they're doing the experiments, thanks God. So we were fine, we were not mixing it too much, but it's a very good question, and something that's becoming really important for all neuroscience, the time of doing experiments.
Pessoal, mais alguma pergunta? Não? Então, gostaria de novamente agradecer a professora Katarina pela disponibilidade, pela excelente apresentação, foi muito interessante, acho que é um tema super importante, né, que, na verdade, acho que essa questão de fazer, né, isso que você comentou, de trazer isso para a população, né, é um trabalho super aplicável, né, e eu acho que uma relevância, né, muito, muito grande. Então, foi um prazer tê-la aqui conosco, né, espero que a gente possa se encontrar em Natal ou aqui em Florianópolis, né, quando toda essa pandemia aí se resolver. Não sei se o Matheus quer falar alguma coisa, Matheus?
Eu gostaria de agradecer também. Eu gostaria de agradecer também ao aceite do convite de trazer o seu trabalho até nós, de participar desse período de integração, que embora todo mundo esteja afastado em lugares diferentes, a gente pode aproveitar pelo menos para tentar aproximar mais as pessoas, né? Você foi uma das pessoas que nos proporcionou isso. Então, só temos a agradecer, em nome de todos os discentes. Muito obrigado.
E muito obrigada a vocês pelo convite, adorei participar.